probably all seen that ADCs have gone from very niche to necessary over the last number of years, and are now, thankfully, firmly embedding themselves into routine clinical practice. But in order to maintain this exponential growth in the sector, we feel that you need to design these molecules beyond the conventional design. We need to think about how do we design these molecules to reach the hardest to hit tumours, the ones where there are no ideal therapeutic targets, or where there's very complex disease biology leading to a very heterogeneous tumour environment. And that is what we're aiming to do. So at BivicTriX, we focus on developing next generation bispecific ADCs. So we moved away from the sort of simplistic targeting. We identified proprietary combinations of tumour antigens that have a tumour-specific co-expression pattern that you don't find on healthy tissues. So through this, we can unmask the tumour cells, and we can ensure that we pinpoint the delivery of our payloads. But we actually go even further here. We've actually found that we can use these unique co-expression phenotypes to identify the causative cells in a tumour, the ones that are linked to the disease mechanism leading to overall shorter overall survival and relapse. And that's a common feature of our two internal programs at the moment. We couple this with our site-specific payload attachment technology, so we get homogeneous product. We've looked at innovations in the conjugation chemistry as well, so we get reduced payload loss. And then importantly, the last box, we can design these drugs so that we get vastly improved internalization. We see synergistic cell killing, and we can kill not just the high antigen expressing tumour cells, but also the low and ultra low expressing tumour cells. And this is what we call a Bi-Cygni ADC. So we start by identifying a clinically validated ADC target, one that has known clinical challenges when associated with the traditional ADC design. We then plug this into our target discovery model, and we look for the optimized pairing for this target. And we're looking for a unique tumour-selective co-expression phenotype that we don't find on healthy cells. And we do this using a multi-layered approach. We use single-cell transcriptomics, proteomics, and then we do functional live cell screening on primary patient samples, looking at expression and proximity. And we are now amassing a growing library of the proprietary antigen combinations to treat several issues that we currently see in the clinic today. But further to this, we were one of the first, if not the first, pioneers of the bispecific ADC movement. We actually predicted this trend before ADCs took off in 2019. So we've been growing our expertise and our knowledge in this area over many years. And we've combined this to produce a very robust and sophisticated internal functional screening platform, where we start by using our plug-and-play proprietary molecule initially that gives us information on the affinity requirements and the epitope accessibility requirements, so we can go straight into an informed target antibody discovery campaign. And then we screen everything in final format as a bispecific ADC. So we don't believe, we've looked at lots of different approaches, we don't believe in the modular designs and starting with monospecifics, and we have reasons for that as well. And this gives us a very expeditious route to lead generation. And we can tailor this design to what we call an and-gated bispecific approach, or what we call an and/or gated bispecific approach, depending on the clinical need that we're trying to solve. So we're using this knowledge to build a growing library of Bi-Cygni ADCs. In the solid tumour space, our lead indication is ovarian cancer, and here we are looking to overcome the clinical challenges associated with B7-H4 targeting, and particularly the need for very high levels of expression in order to see meaningful clinical efficacy. We actually have data to show that expression of B7-H4 is causally linked to ovarian cancer, so there is a requirement to remove not just the high, but also the low B7-H4 expressing cells. And so we've identified a proprietary cancer-specific antigen combination and designed, let's say, what we call an and/or gated bispecific ADC, which allows us to not just remove those high expressing tumour cells, but also be able to effectively eliminate low and ultra low dual expressing cells at doses that don't harm healthy cells. And we've recently resulted in some data, preclinical model of ovarian cancer ran head-to-head with 2 clinical comparators, and we were able to significantly outcompete those 2 clinical comparators, and we saw tumour regressions of over 84%. And further to that, in a mouse that actually didn't respond to AstraZeneca's Topo1 based ADC, we gave a single dose of BVX-002, and we were able to show that we not only stopped the exponential growth of the tumour, but we also resulted in over 77% tumour regressions in 21 days from that single dose. And then looking more in the heme space, we also have an asset that's targeting a newly emerged and distinct AML subtype called CD7 positive AML. The likes of MD Anderson have recently produced data on showing that this subtype is linked to shorter overall survival and treatment failures, particularly using Veneza. And here we've identified a leukemic specific combination of targets, and we've designed an AND-gated bispecific ADC, so we can effectively eliminate these problematic leukemic cells at doses that are not toxic to healthy myeloid or lymphoid cells. And we have PDX data and humanized mouse data to show this, and we have very broad protection on this antigen pairing with patents now granted globally as well. And because of the excitement in the data and the excitement in the that we are attracting a top-tier leadership team. So Peter Baby on the right-hand side has got over 2 decades of experience in ADC development from large pharma, and he's currently co-founder and CSO of a company called Atavia. They've raised over $250 million from the likes of Frazier Life Sciences in the last 2 years alone. And then Adam Freund as well in the bottom corner here. Adam's CEO and founder of a company called ADCs Therapeutics, backed by Global Ventures and A16Z. And he's really working at the forefront of moving ADCs outside of oncology and looking at their application in inflammatory diseases. And then our chairman, Michael Kaufman, has raised over $1 billion in his prior company, Karyopharm, and he has 3 successful drug approvals under his belt. So that was a whistle-stop tour, but obviously please do get in touch if you'd like to hear any more. I'd be happy to obviously share the data on our programs, and my contact details are on the screen. And thank you very much for listening.